Hello students, our today's topic is protein kinases, and today we will discuss structure, types and function of protein kinases. The eukaryotic protein kinases are defined as enzymes that use the gamma-phosphate of adenosine triphosphate, means ATP, to phosphorylate serine, threonine or tyrosine residues in proteins, because phosphorylation mainly occurs at the serine, threonine and tyrosine residues. Now, where this gamma phosphate of adenosine triphosphate is present on ATP. Look at this diagram. This is a structure of ATP. And ATP is composed of an adenine ring. This is an adenine ring, ribose sugar, and three phosphate groups, 1, 2, and 3. The phosphate group, these groups of phosphate group are usually called alpha phosphate group, beta phosphate group, and gamma phosphate group. Gamma phosphate group is the primary phosphate group on the ATP molecules that is hydrolyzed when the energy is needed to drive anabolic reactions. Basically, this gamma phosphate group is present, means typically located the farthest from the ribose sugar. You can see here, this is ribose sugar and this is gamma phosphate group. This gamma phosphate group has higher energy of hydrolysis than either that of the alpha and the beta phosphate, right? When the first plant genome, means Arabidopsis thaliana genome, was sequenced, a surprising number of protein kinases, over 1000, were identified. The first plant protein kinase sequence were identified in pea and in rice through the use of degenerate primers. Now, structure of protein kinases. This is a structure of protein kinases. Protein kinases share a similar three-dimensional catalytic domain. This is catalytic domain. Catalytic domain consists of 250 to 300 amino acids and composed of a larger, generally alpha-helical C-terminal subdomains and a smaller, usually beta-sheet N-terminal subdomain. This is N-terminus, this is C-terminus, and this is catalytic domain. Now, this N and C-terminus are linked by peptide scaffold. Forming a deep groove enables the binding of peptide, substrate, and an ATP molecule, right? This is ATP binding site of catalytic domain, and this is substrate binding site. Now, ATP binding region rotates into on and off conformation depending on ATP binding and the activation state of the enzyme. Next, types of protein kinases. Protein kinases are classified into 5 major groups according to the amino acid residue that they phosphorylate, right? Including serine threonine protein kinases, abbreviated as STPKs, tyrosine protein kinases, TKs, histidine specific kinases, dual specificity protein kinases, and aspartic acid, glutamic acid specific protein kinases, right? First, serine threonine protein kinases. Serine threonine protein kinase is a large family of protein kinases, mainly includes cyclin-dependent kinase, mitogen-activated protein kinase D, nattokinase, DNA-dependent protein kinase, aurora protein kinases, and pancreatic kininogenase, right? So first we'll start with the first type of serine threonine protein kinases, that is cyclin-dependent kinase. Cyclin-dependent kinases are abbreviated as CDKs. They play a central role in cell cycle regulation network. And its main biological role is to regulate different phases of the cell cycle from G1S, G2 to M phase, and means to complete the whole cycle, right? The CDKs subunit need to be combined with corresponding cyclin to activate. That's why cyclin-dependent kinase. Activated CDKs exhibit protein kinase activity that phosphorylate different substrate proteins, thereby initiating or regulating the cell cycle, right? The substrate activated by CDKs mainly include retinal glioma protein, tumor suppressor gene, that is P107 and P103, etc. Right? Which have important functions of promoting cell cycle phase transition, initiating DNA synthesis, running cell division, and promoting cell cycle operation. CDKs are also involved in regulation of transcription, mRNA processing, and differentiation of neural cells. Right? Next is mitogen activated protein kinases, abbreviated as MAPKs. MAPKs are essential components for controlling embryogenesis, cell differentiation, cell proliferation, and cell death. They're also involved in directing cellular response to various stimuli such as mitogen, osmotic stress, heat shock, and pro-inflammatory cytokinin. Right? Mammals express at least 4 distinctly regulated groups of MAPKs. First is extracellular signal-related kinase. Second is N-terminal kinase. Third, P38 protein. And the fourth one is ERK5. Next is protein kinase D, abbreviated as PKD. It is involved in regulation of cellular function such as cell Golgi reverse membrane transport, cell growth, proliferation, migration, differentiation, and apoptosis. PKD protects the myocardium and reduces the damage of cardiomyocytes induced by calcium overload. Right? It affects the transport of transferrin receptor, means TFR, and low density receptor related protein, LRP, in hippocampal neurons. PKD is also involved in regulation of the release of inflammatory factors. Right? Next type of serine threonine protein kinases is pancreatic kininogenase, abbreviated as PKs. PKs can improve sensory or motor nerve conduction velocity through kinin, right? Inhibit platelet aggregation, prevent blood clot formation, dilate blood vessels, enhance microcirculation, and reduce ischemia and hypoxia. PKs has the same efficacy as prostaglandin E1, means PGE1, in improving pathological changes of diabetic peripheral neuropathy and improving neurological function in patients with type 2 diabetes. Therefore, PKs will become a potential new drug for the treatment of diabetic neuropathy. Right? Next type of protein kinases is tyrosine kinases. Tyrosine kinases are classified into 3 types: non-receptor tyrosine protein kinases, receptor tyrosine kinases, and nuclear tyrosine protein kinase. First is non-receptor tyrosine protein kinases, abbreviated as NRTKs, right? Non-receptor tyrosine protein kinases are divided into 11 families with at least 30 members. They mediate signal transduction of various growth factor receptors, cytokine receptors, lymphocyte, antigen receptors, and adhesion molecule integrins. NITKs mainly including SRC kinase family, JAK kinase family, SKY, and ZAP70 family. Now, SRC kinase family, this is a product of proto-neogene CSRC, involved in antigen receptors, cytokine receptors, and integrin-mediated transmembrane signaling. SRC kinase family, right? Second, JAK kinase family, including JAK1, JAK2, JAK3, and TYK2. They mainly mediate transmembrane signal transduction of cytokine receptor. And the last one, SKY, ZAP70 family, including SKY and zeta gene associated protein 70. It mediates signal transduction of lymphocyte antigen, uh, means lymphocyte antigen receptors and certain cytokine receptors. And it is also important in lymphocyte differentiation, development, and activation. Right? Now, next, receptor tyrosine kinase. Receptor tyrosine kinase is a cell surface receptor that has also a tyrosine kinase activity. A signal binding domain of receptor tyrosine kinase is on the cell surface, while the tyrosine kinase enzymatic activity reside in the cytoplasmic part of the protein. Right? A transmembrane alpha helix connects these two regions of the receptor. Right? Receptor tyrosine kinases are essential components of signal transduction pathways that mediate cell-to-cell communication. These signal pass transmembrane receptors, which bind the polypeptide ligands, mainly growth factors, that play a key role in processes such as cellular growth, differentiation, metabolism, and motility. Right? Approximately 20 different RTK classes have been identified. Next, nuclear tyrosine protein kinase. Protein tyrosine phosphorylation plays an important role in the transduction of extracellular signals. The prototypical protein kinase are localized at plasma membrane. Just listen. Prototypical protein tyrosine kinase mainly are localized at the plasma membrane and are coupled to receptors that bind extracellular factors. Thus, protein tyrosine phosphorylation was previously thought to occur only in the cytoplasm. However, several cytoplasmic tyrosine kinases have recently been found to enter the nucleus and means resident nuclear proteins such as catalytic subunit of RNA polymerase 2 have been found to be phosphorylated on tyrosine. Nuclear tyrosine kinases may participate in the regulation or transcription, the cell cycle, and possibility on other nuclear processes. Right? Next class of protein kinases is histidine-specific protein kinases. Histidine protein kinases is a kinase that phosphorylates histidine in a substrate protein, mainly including two-component histidine protein kinase, means it can regulate the response to environmental stimuli, and two-component mammalian histidine protein kinase. One is branch chain alpha ketoacid dehydrogenase kinase and pyruvate dehydrogenase kinase. Next is dual specificity protein kinases. Dual specificity kinases, as its name indicates, phosphorylate their substrate on serine, threonine, and tyrosine residues. They intervene in the regulation of cell growth, differentiation, and apoptosis. Dual specificity kinases include at least members of superfamily of mitogen activated protein kinases as well as those of the family of glycogen synthase kinases. Right? And the last type of protein kinases is aspartic acid glutamic acid specific protein kinases. Aspartic acid glutamic acid specific protein kinases are a category of protein kinases that can phosphorylate aspartate and glutamate. Right? So this is all about the structure, type, and function of protein kinases. If you have any question, just write down in the comment section so that I can answer. Right? Thank you so much.